All right, so we're picking back up here, and once again, we're talking about what happens with those electrons that were generated from rather the electron carriers that were generated from the TCA cycle and also glycolysis. Remember, those electrons just can't sit there; they have to go somewhere. So we were talking about how we're going to utilize respiration. With respiration, the electrons from these electron carriers are going to be donated to an electron transport system or electron transport chain. As these electrons move through the electron transport system or chain, the energy in them, because electrons are like in constant motion, so they have high amounts of energy. So as these electrons move through the components of the electron transport chain, that energy that they're giving off is harnessed, so to speak, in order to move hydrogen ions in a given direction. Now, hydrogen ion is also called a proton. So we say that the electrons, their energy is used to generate a proton motive force or simply a proton gradient. And then that proton gradient is going to be used in order to generate ATP. We'll see how that all plays out here shortly. But once again, even though we've donated the electrons to an electron transport chain or system, they still have to go somewhere. So at the end of this process, there has to be a final electron acceptor. It really kind of comes down to two major categories. First of all, you can have oxygen. If oxygen is your final electron acceptor, you produce water as a byproduct. We call it simply metabolic water. And in that situation, since you are using oxygen, you would have aerobic respiration. There are other electron acceptors as well. If you use one of the other ones, you accomplish anaerobic respiration, and the byproducts that you yield are going to be various depending upon that particular electron acceptor outside of oxygen. Here we kind of have a little diagram to kind of give you some perspective. Once again, the process of respiration is going to produce a lot more ATP than when compared to fermentation. Remember, fermentation was a way to donate or recycle NAD plus because we're going to donate the electrons from NADH to another organic molecule. The problem is it really doesn't produce any ATP. Notice you're only getting your acids and alcohols here. So when we accomplish fermentation, we don't get that big payoff because we never give our electrons to an electron transport system or chain. But remember, when we are going to accomplish respiration, we get that middle step, the TCA cycle, and then we get into the electron transport chain or system. Aerobic respiration will, once again, as I mentioned a moment ago, yield water as a byproduct. We do recycle our electron carrier so we can continue adding electrons to them. But then look at the yield here of ATP, roughly around 34. Now that's a maximal theoretical yield. Do we always get that? No, but you could. Combine that with the 2 GTP that we converted into ATP from the TCA cycle, along with the 2 net ATP from glycolysis, and you can maximally yield 38 ATP theoretically from one glucose molecule. Anaerobic respiration also recycles your electron carriers. As I told you, you'll get various products depending upon which final electron acceptor you use, but all in all, the anaerobic respiration usually yields fewer ATP. It really relates to the electrical potential of the final electron acceptor. Oxygen has a greater redox potential than any other final electron acceptor. The greater the redox potential, the more you can pull the energy out of the electrons. I'll show you a little diagram maybe to relate that in a couple of slides here. Aerobic respiration, regardless of where it takes place, needs a membrane. So let's think about this for a moment. Aerobic respiration is accomplished by both eukaryotic and prokaryotic cells. When we look at eukaryotic cells, they're going to utilize, zoom in a little bit here, their mitochondria. The mitochondria are able to accomplish aerobic respiration. If you don't remember the basic structure of a mitochondrion, it has an outer membrane and an inner membrane with these folds, cristae there. The components of the electron transport chain or system in a prokaryotic cell are located in that inner membrane. So notice, we'll zoom in back over here. The inner membrane is where we're going to house these components. Now, I don't go through and ask you guys to remember this, but you see complex 1, 2, 3, and also 4. So there are 4 complexes in that mitochondrial membrane. And when the electrons are donated, notice you see NADH donating electrons here, the electrons are going to move through the components in a sequence. And as a result, you pump hydrogen ions into the space between the inner and outer membranes. It's called the intermembrane space. This is your proton gradient or proton motive force. It's building up protons here in this intermembrane space. There is a little bit of a caveat. If you look at FADH2, it doesn't donate its electrons at complex 1. It typically donates its electrons at complex 2, so you're not going to generate as many protons moving into that intermembrane space. So from FADH2, we don't get as much of an energy yield. It's about where it donates its electrons. It donates its electrons at complex 2 instead of complex 1. So you actually get a little bit more energy from NADH than you do from FADH2. But this slide is meant to make you, or hopefully get you to realize that for eukaryotic cells, you have to, or rather, you're going to see this process play out within the mitochondria. So the question becomes, what about our prokaryotic bacteria and archaea? How are they going to possibly be able to accomplish aerobic respiration? Because they're prokaryotic. They don't have any organelles like mitochondria. Well, they do have a membrane. So instead of using the mitochondrial membrane, what they do is they substitute their own plasma membranes. So it would be a similar setup that you would see, zoom back in here, in a prokaryotic cell. Complexes 1, 2, 3, and 4. The major difference is where it's located. Instead of being in the mitochondrial inner membrane, because prokaryotic cells don't have mitochondria, these components would be in their plasma membrane. And remember the structure of those bacteria, especially, there is that little periplasmic space. So all these protons would be building up in the periplasmic space in order to generate your proton motive force or your proton gradient. So the periplasmic space, along with the cell or plasma membrane, that's where we're going to see this process play out when it comes to prokaryotic cells. As it says here, the electrons are passed down the line, so each complex is harnessing the energy in order to move hydrogen ions or protons. And ultimately at the end, you'll see if it's aerobic respiration, the oxygen will accept those electrons. And I'll just zoom in over here. You can see here's your oxygen accepting the electrons to become water as a byproduct of aerobic respiration. As I told you before, this is a slide I was going to show you. Oxygen does give the best result when it comes to respiration. So aerobic respiration gives you the most energy because look at the redox potential of oxygen. It is the most positive or the highest redox potential. So you get the most energy because you can accept electrons all the way down the line at complex 4. All the others usually accept the electrons at an earlier point. So because of that, you don't get as much of a payoff, so to speak. Notice all of these are going to be used for anaerobic respiration, and these are molecules that are, well, not oxygen. So you can see iron, for instance. We can talk about nitrate, fumarate, which was one of those precursor molecules we saw in the TCA cycle, sulfate, carbon dioxide, sulfur. So there's a multitude of other electron acceptors. The thing is, they don't allow you to generate as much ATP. Any of those other electrons, or rather, any of those other electron acceptors, if they're taking on the electrons, we refer to it as, once again, anaerobic respiration. We don't get as much ATP because the oxygen has the greatest redox potential. You don't have to remember the number, but the oxygen having the greatest redox potential, it means that it can accept the electrons at a further point down the line, and that means that you're able to, well, move more protons to generate more ATP. All the other electron acceptors have to accept the electrons at an earlier stage or earlier point, and because of that, you won't generate as much ATP because you're not going to move as many protons into the intermembrane space or into the periplasmic space if we're talking about bacterial cells. There is the possibility that some microbes can do both. Actually, we have a name for them. If you can do both, we usually refer to you, there's several different categories depending upon the degree to which you do it, but usually we refer to you as a facultative anaerobe. Let me see if we maybe have that slide. There we go. See these? Facultative anaerobes, this term here, we'll zoom in real quick. These are microbes that can accomplish anaerobic respiration, but they grow best when they're carrying out aerobic respiration. So there's some microbes that can do both. They can do anaerobic and aerobic respiration. So you don't have to be exclusively one or the other. These facultative anaerobes, they're going to grow best when they have oxygen present. Why? Because they can accomplish aerobic respiration. Remember, why would they grow best? Because they're going to get more ATP. They have more energy to grow. But if you take away their oxygen and they can't use O2 as a final electron acceptor, they can switch over and accomplish anaerobic respiration. They will grow slower when they're doing their anaerobic respiration because they don't make as much ATP. E. coli is a classic example of a facultative anaerobe. It can accomplish both aerobic respiration and also anaerobic respiration. I just put this in here so you can see how that process plays out. You don't have to go in and memorize the different complexes and all that kind of stuff. That's not what we're going to get into here. But once again, these organisms that are facultative anaerobes go slower under anaerobic respiration because you just don't make as much energy. So let's go back and talk a little bit about that proton motive force that I mentioned to you. The proton motive force is what you get when you pump all those hydrogen ions into the periplasmic space or that inner membrane space of the mitochondria. Notice you build up all these protons, and guess what? It can be used to make ATP. It can be used to allow for the flagellum of a bacterial cell to spin. It can be used to move nutrients. So there's a lot of things that can be accomplished by this proton motive force. But in the case of making ATP specifically, because that's what we've been focusing on, that's what we've been talking about. Well, as you build up all these protons in that space, nature takes over. Nature wants equal concentrations across both surfaces. So we start to see diffusion. Well, we only allow those protons to diffuse back into to the opposite side of the membrane in one way. It's by moving through an enzyme called ATP synthase. And here's where your payoff comes in. As these protons diffuse back into back across the membrane, I'll say it that way, through ATP synthase uses that energy of motion, the movement of those hydrogen ions or protons, in order to phosphorylate ADP. ADP picks up a phosphate group and we make our ATP. So there's our payout. We have to have the ATP synthase enzyme as well. So ATP synthase is used in order to produce our ATP. The protons from our proton motive force are going to diffuse or move back through. We're not going to worry about all the various subunits, who's spinning, who's doing what. That's a little beyond the scope of this course, I really believe. But as those protons move, I need you to appreciate the fact that as they diffuse, we're going to use the energy from that proton motive force to phosphorylate our ADP and form ATP. And then there's our big payoff. We move so many protons under aerobic conditions, at least, that we should be able to make close to 34 ATP. Once again, that's a theoretical maximal yield. Does it always come out at the theoretical maximum? No, it does not. But you'll have that proton motive force generating your ATP as those protons move through ATP synthase. Now, we've looked a lot at how microbes are able to generate energy from the standpoint of breaking down glucose. But there are many other carbohydrates that can be utilized other than glucose. The reality is simply that microbe has to have the enzymatic machinery, so to speak, in order to break down that particular polysaccharide. So can you use other sugars, other polysaccharides to generate ATP? Absolutely. All you have to do is be able to break them down. Now, that's the key. Not all microbes can break down every sugar. For instance, some can break down lactose. Some can't break down lactose. You have to have lactase in order to do that. So you have lactase, you can use lactose as a potential sugar in order to gain your energy. So this slide is really addressing the fact that you can utilize other molecules besides just glucose. Why do we look at glucose? Because it's a fundamental molecule that almost every organism has the ability to degrade in order to generate ATP and metabolic precursors. Not only can you just use other sugars, you can also use other macromolecules, proteins and amino acids. Well, I can give you a human example. If you're starving, your body breaks down muscles in order to generate energy from the amino acids that are making up the proteins within your muscles. So proteases are enzymes that break down proteins. If you break those up, you can get your individual amino acids. Then You just have to do a little bit of manipulation. If you remove the amino group, which contains the nitrogen, that gives you an organic acid. It's actually called carboxylic acid, and those carboxylic acids can be used in the TCA cycle. So proteins can even be metabolized for sources of energy as well as metabolic precursors. And then absolutely lipids. Lipids are just carbon, hydrogen, and oxygen primarily. You can attach some different groups if you like and make them slightly different. But if you look at a carbohydrate, it's carbon, hydrogen, and oxygen. They're just attached a little bit differently. So in order to use lipases, we can do a couple of things here. We can separate the fatty acids. You think about phospholipids. They have a phosphate head, fatty acid tails. Strip off the fatty acids, you've got your lipids. You've got various triglycerides, which are like the little microscopic fats. If you beta oxidize them, in other words, take away some electrons, break a couple of bonds, you can make the fatty acids into small little chunks, quote unquote. They're literally little chunks of carbons, hydrogen, and oxygen, just like what you would have if you broke up large polysaccharides. And when you do that, you can then funnel that into the tricarboxylic acid, and it can continue on for respiration. So these last couple of slides are really just meant to tell you this. Don't assume, because it's not true, that carbohydrates or glucose, more specifically, is your only source of energy. You can get energy and precursor molecules from other sugars, or rather proteins, and also lipids. So it is possible to gain what you need from other sources. All right, so that's pretty much the end of the respiration information. A little bit shorter video here. In the next, we'll kind of move on and we'll talk a little bit about photosynthesis, as you can see here.